Alright guys, we're going to talk about biochemistry today, proteins and nucleic acids. Make sure you're filling out your handout as you go along, and we will get it glued on page 25 and 26 when we've completed the video lecture. So we're going to talk about macromolecules again. Remember the term macro meaning large. So these are large molecules that we utilize in our bodies. So the first one we are going to talk about is protein. Remember, monomer meaning the smallest unit that makes up that macromolecule. So the monomer of a protein are called amino acids. Our body has 20 different types of amino acids that we will combine using our DNA sequence to create a protein. The polymer, or the finished product, is a protein, or also called a polypeptide. The structure of the protein, we have a central carbon atom with hydrogen, amine, carboxyl, and R groups. That R group is what basically determines the structure or the final product, which protein is going to be. Well, the uses of protein. Well, we use proteins to basically make every structure in our body. Your hair, your nails, your skin, the bones, your muscle groups, your organs, your blood, the veins that carry your blood, all of those are based off of proteins. Protein is also an emergency energy. If we've used up the carbohydrates and the lipids, our body will resort to the last thing, and that would be breaking down muscle tissue and organs to provide energy. And the only time this would be needed is if you're stranded somewhere for days without anything to eat and you haven't replenished your carbohydrates and lipid storage, so therefore resort to emergency energy to keep you alive. Examples of protein would be your skin, insulin, and also enzymes, your muscles, your hair, your bones, your large your biceps, your quads, glutes, organs, all of those are protein. So we have 4 different types of proteins. We have structural protein, which would give us our faces, our lips, hair, nails, skin. We have contractile proteins. Those are your muscle groups. When your quads fire and they help you move and run, your biceps. And then we have storage proteins. You can find those in a lot of, I guess, for example, peanuts. And we have our transport proteins that help move things around. In this case, image, we have red blood cells going through a capillary. So proteins are building blocks of structures called amino acids. Amino acids are what we combine to create proteins. Proteins are what your DNA codes to make. So all those A's, T's, G's, and C's on your DNA combine in certain groups to form certain amino acids. A peptide bond forms between amino acids by dehydration synthesis. So dehydration synthesis is the building up of large molecules by removing water molecules. Here we have pictures or examples of the protein, and you can see our R group. Here's our central carbon, here's our amino group, our carboxyl group, and then our R group. And this R group helps determine the shape. And the shape of a protein is what determines its function as well. Proteins also build enzymes. An enzyme is a special protein that speeds chemical reactions. Chemical reactions require a certain activation energy to get started. Enzymes decrease this energy, making reactions occur faster. So here you can see in this chart we have the energy supplied, the energy released. We start at the same point here, but without an enzyme, we require a large portion of energy to complete a process. So we start here, we need all this energy, all this energy, all of this energy to complete to our final step. But if we add in that enzyme and we're doing the same thing, the same task, we add this enzyme in and we need a fraction of the energy to get the same task done. So enzymes speed up a reaction without changing anything. They just make it occur faster. So enzymes are a good thing. It gives it that activation energy to get started, and we don't need this would be wasted if we didn't have an enzyme. So, and enzymes are basically like a lock and key model. Enzymes are not used up by the reaction, but each can only work on one reaction, and we call this enzyme specific. I can't say the word. It's enzyme specific. So enzymes can be used over and over again, but they can only be used with certain things. This is called the lock and key model of enzymes. An enzyme is like a key which can open exactly one lock. If you want to unlock another reaction, you need a different enzyme. Factors which affect enzymes. Temperature. Enzymes, like all proteins, can change shape when exposed to heat or cold. Each has an optimal temperature range. If they change shape, then they're no longer functional or no longer usable. The pH. All enzymes have an optimal range of pH. For example, we have your hydrochloric acid, which is in your stomach. It likes to be at a certain pH. Concentration. Having more enzymes makes the reaction faster. So temperature, pH, and concentration affect how the enzymes function. So here we have an example of an enzyme. Here we have our enzyme. Here we have the active site will only fit to a certain substrate. So this fits in here. Then we call it our enzyme substrate complex. Our substrate will then be broken down into products. This enzyme is now ready to start the process over and over again. So this enzyme is reusable. We have our active site, which fits the specific substrate. When it's combined, it's an enzyme substrate complex, and it breaks the substrate down into products. Now let's go into our nucleic acids and ATP. So we have macromolecules, our nucleic acid. The simple structure or the monomer of a nucleic acid is called a nucleotide. We also have the polymer, which is your nucleic acid, which is when your nucleotides combined. We have a 5-carbon sugar attached to nitrogen base and a phosphate group. So the uses of nucleic acid stores your genetic code. The examples of your nucleic acids are DNA and RNA. So here we have our nucleus containing 23 pairs of chromosomes. Here's what a chromosome looks like. These dark lines represent the genes, and if we stretched our chromosome out, we would find that we have our DNA strand. So this is your double helix of deoxyribonucleic acid, and these rungs are your bases, and they contain the genetic code. So here is a nucleotide. We have our base, our sugar, deoxyribose in this case because it stands for DNA, and here is our phosphate. The phosphate and the sugars make up the sides of DNA. Your bases make up the rungs. So here we have the deoxyribose sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate. Your bases are attached only to the sugar. And these make your genetic code. So here again is our nucleotide. Oops, and some of this didn't show up. Nucleic acids are polymers of nucleotides. Here we have our sugar, our phosphate, and our nitrogen base. We can have adenine, guanine, cytosine, or thymine on DNA, and then we would add uracil for RNA. So again, here's your nucleotide, which is your nucleic acid monomer. Our sugar, which is attached to the base, and then from the sugar we have our phosphate group. The phosphate group and the sugar are the sides. The bases make up the middle of DNA. Here are our bases. We have adenine, guanine, thymine, and cytosine. And here we have, you can see the picture of adenine, guanine, thymine, and cytosine. So the nucleotide or the monomers form long chains called DNA. So here we have our DNA strand. This is the backbone. These are our bases. A nucleotide would be our base, a sugar, and a phosphate. These three pieces make up the nucleotide. Nucleotides are joined by sugars and phosphates on the sides. So two strands of DNA join together to form a double helix. RNA, which is ribonucleic acid. Instead of deoxyribose, we contain ribose sugar. It has an extra hydroxyl group. It has the base uracil instead of thymine. So now let's talk about ATP, which is our primary energy. ATP stands for adenosine triphosphate. Cells use ATP as a rechargeable battery. It is made up of an adenine with 3 phosphates. Lots of energy is stored in the bonds between the second and third phosphate. When this bond is broken, tremendous energy is released. The pieces are then reassembled, storing more energy for another use. So this is basically a rechargeable battery. All of our energy comes from ATP. Every time we break the bond between the second and third phosphate, we're releasing a tremendous amount of energy. We can then replace that third phosphate back onto the 2 phosphates, and we'll start all over again. So make sure that you're able to explain the ATP-ADP process. ATP has 3 phosphate groups. The energy is stored in the bond. When one phosphate group breaks off, it releases energy in forming ADP, meaning adenosine diphosphate. The addition of one phosphate to ATP or ADP forms ATP, and the process continues over and over. Here we have our macromolecules. Remember carbohydrates, which is our short-term energy storage, also plant structure, and then we have our monomer is a monosaccharide. Lipids, long-term energy storage, fats, and hormones. Proteins, enzyme structures, storage, contraction, transport, and their monomer is amino acid. Nucleic acids, the information storage, your DNA and RNA, and the monomer is a nucleotide.